My name is Maria Kholmanskikh, and today I will be presenting my completed project, PLA as a novel method for assessing Rho GTPase activity. Rho GTPases are a subfamily in the ROS superfamily of molecular switches. Significant work has been done on the founding member of the Rho subfamily, Rho A, but much less is known about its vertebrate-specific paralog, Rho C, which is 92% identical by amino acid sequence. The Rho GTPase my project is focusing on is Rho C, which is a small Rho GTPase that acts as a molecular switch that regulates various cellular processes by regulating the cytoskeleton and gene expression. By acting upon cytoskeleton and gene expression, Rho GTPases regulate several essential cellular functions: morphology, motility, and division. Dysregulation of the Rho pathway causes many human diseases, including cancer, neurological disorders, disorders of the immune system and skin, and cardiovascular disease. Rho GTPases are molecular switches that go from the on GTP-bound state, where they can bind to their downstream effectors that regulate cellular function, to off or GDP-bound state. The rate of GTP hydrolysis by Rho proteins is very slow, and that's why hydrolysis is facilitated by GAP, GTPase activating protein, molecules that facilitate the rapid inactivation. Rho GTPases have a high affinity for GDP, and despite tenfold excess in GTP concentration, the rate of exchange would proceed very slowly, and that's where GEFs, or GTPase exchange factors, come in. When bound to Rho, they change its conformation and lower its affinity for GDP, and rapid exchange for GTP occurs. For example, a blood serum component, LPA, activates through transmembrane receptor Rho proteins, which in turn activate a variety of downstream effectors, such as ROC1. While methods do exist to measure Rho GTPase activity, they either only measure a single GTPase and rely on overexpression of exogenous tagged proteins, such as biosensors, where Rho GTPase and effectors are attached to fluorescent proteins, and the biocomplementation assay, which only looks at quantity of activity instead of luminescence. Or they lack cellular and subcellular resolution, such as the pull-down assay, where the cell or tissue are lysed. Most importantly, no existing method measures interactions with specific individual effectors. PLA, on the other hand, is specific and efficient. So how does it work? The technique requires two antibodies of different species to recognize the two proteins of interest, which is followed by the addition of secondary antibodies attached to complementary oligonucleotides. When in proximity, or less than 40 nanometers apart, oligonucleotides will hybridize and can be ligated into a contiguous stretch of DNA. This DNA can then be amplified by DNA polymerase with fluorescently labeled oligonucleotides, or nucleotides, resulting in an accumulation of fluorescent spots in areas of high levels of protein-protein interactions. So, can proximity ligation assay, or PLA, help to determine patterns of activation of Rho C and which effector is downstream of Rho C in a particular process? In other words, will PLA be effective in, first of all, measuring Rho C activity, and second of all, in assessment of effector specificity with subcellular resolution? We use small hairpin RNA, or shRNA, against Rho C mRNA to knock down the level of Rho C protein. So in epithelial or fibroblast cells with less Rho C compared to control cells, we performed immunofluorescence labeling of Rho C. The data was then collected and signal intensity was analyzed using ImageJ. And this is an example of what analysis using ImageJ would look like, with finding the area, and that gives us the signal intensity. This was followed by PLA optimization. And after the specificity was tested, PLA was optimized to measure Rho C interactions with their effector molecules. We serum starved cells followed by PLA to measure the Rho C to ROC1 interaction. And again, this was followed by image J analysis. When we tested Rho A antibody in cells with reduced Rho A, we didn't see a significantly decreased signal, meaning the binding wasn't specific, even though we knew it should have been. In the case of Rho C antibody, we did see a significant decrease in cells with reduced Rho C, but for some reason we saw the same significant decrease when we used cells with reduced Rho A. As you can see here, there was no significant decrease, but there was a significant decrease in these two. Because we know that the shRNAs work, the issue must have been with either the antibody or the staining method. One of the ways to try to adjust any method is to adjust the method of fixation. That's why instead of the most commonly used fixative or of paraformaldehyde, we opted to use an alternative method of fixation, glyoxal. They do a different degree of cross-linking, and the resulting protein modification by paraformaldehyde and glyoxal is different, and you end up with different modifications of the protein. So this is an example of glyoxal and paraformaldehyde, and they do a different degree of cross-linking, resulting in different modifications. And this did end up working for the Rho C antibody. Here we test, this is the specific anti-Rho C antibodies, and they produced reduced signal in cells with reduced Rho C, but not in cells with reduced Rho A. So this validated the antibody. The blue is the control, the orange is the reduced levels of Rho A, and the green is the reduced levels of Rho C. Again, as you can see, there was no significant decrease in signaling when Rho A was reduced, but there was a significant decrease in the level of signaling when Rho C was reduced, meaning that the antibody specifically binds to Rho C. This is the immunofluorescence staining of control cells, cells with reduced Rho A, and cells with reduced Rho C. When comparing transduced and non-transduced in control and reduced Rho A cells, the amount of signaling is similar. When comparing transduced and non-transduced cells with reduced Rho C, the amount of signaling is clearly less. The left is showing the cell that is transduced over here, and the right is showing the level of signaling intensity. As you can see, it is significantly less. This was followed by PLA validation. So after the specificity is tested, PLA was optimized to measure Rho C interactions with its effector molecules. We serum starved cells with using LPA, which is a component of serum and is a potent activator of Rho and Rho C, followed by PLA to measure Rho C with ROC1 interaction. The serum-free cells had reduced levels of Rho C and produced significantly lower levels of signaling. There was actually a 7-fold difference between the serum-free and the control. PLA signal in cells in control or serum-free media, and when compared to the controls, the serum-free has little fluorescence, showing that PLA accurately assesses Rho C activity. The significant PLA signal increase under conditions promoting Rho-GTP interaction with ROC1 validated PLA's utility in monitoring the Rho-GTPase activity. Our objective was achieved. The antibody was validating, showing that the binding between the antibody and Rho C is specific, and the quantity of signaling is accurate. PLA was optimized to measure the interaction between Rho C and its effector ROC1. By using PLA to look at the downstream effect of Rho C, it is possible to find therapeutic targets for diseases that are caused by the dysfunction of Rho-GTPases and their pathways, and neurological disorders, including Alzheimer's. It is also a major advancement in the methodology of measuring activity of Rho proteins. Now, we can combine the advantages of a pull-down assay, which looks at endogenous proteins and can distinguish between isoforms, with the advantages of biosensors assay, which provide cellular resolution. And it also brings in a whole new dimension that wasn't there before, which is to look at the interaction of a Rho protein and a specific effector molecule. For future directions of my research, we still have not confirmed Rho A, so we only looked at Rho C, and we would also like to test additional effector molecules, including G-formins and PKN. Also, amyloid beta activates the Rho pathway, but we would like to look at whether that is just Rho A, just Rho C, or maybe both. And also, downstream of Rho A row A and row C, we would like to look at which specific effectors are activated.
2-minute warning.
I'd like to acknowledge my mentor, Dr. Ross, my parents, Dr. Kholmanskikh and Mrs. Vidianska, and my teachers, Mr. Lewis and Mr. Pratt. Thank you.